So thank you very much for allowing me to speak about what we're doing in trying to develop this precision medicine approach using ALS as an exemplar, but I believe that this could be used for other diseases as well. So I'm going to talk mainly about ALS and the challenges that we encounter, but I think it's, as I said, it's an exemplar for other rare diseases. ALS is a rare disease, but it is the commonest neurodegenerative condition of midlife. So that the lifetime risk is about 1 in 300. It's different for men and for women. It's commoner in men than it is in women. So the overall prevalent incidence is about 3 to 4 per 100,000, and the prevalence is about 6 to 10 per 100,000. And obviously the prevalence being just over twice the incidence implies that the condition has a short life expectancy, that the disease trajectory is short, because the incidence is not that different from MS, but obviously those patients live for a long time. We know that it's a complex genetic disease, and we know that a proportion of patients with ALS, depending on how you count it, have a family history of either ALS or FTD or possibly other neuropsychiatric disorders as well. That probably tells us something about the disease. The life expectancy is less than 3 years from first symptom in over 70% of patients. And unfortunately, that hasn't changed over the last 30 years. The diagnostic delay for most patients is about a third of their disease journey. And that's because the disease presents in a way that often can be mistaken for other conditions, such as a foot drop being mistaken for degenerative disc disease, or a dysarthria being mistaken for vascular disease. So often patients will present to their GP with these relatively non-specific symptoms, and it will take some time for the GP to recognize that this is atypical. And on an average, patients with ALS see 4 to 5 different specialists before a definitive diagnosis is made. And so we've done some work on this. Seeing a neurologist as one of those 4 to 5 is not necessarily speeding up the diagnosis. And there's a further delay then in referring patients to specialist clinics. It's about another 3 months. So about half of the patient journey is in the diagnostic or pre-diagnostic phase. So we know that ALS is a complicated disease as well. So we always thought of ALS as a condition involving the motor system, the corticospinal tract, and the anterior horn cell. And the other name for ALS sometimes is anterior horn cell disease, implying that this is a disease of the motor nerves from the time that they are in the anterior part of the spinal cord, and then trajectory out into muscle. But more recently, in the last 10 or 15 years, and our group in Ireland actually has been quite important in defining this, we know that this is a much more complicated disease, and there are many extramotor features. And we can divide these into different circuits that are involved. So, for example, the sensory motor and premotor circuit, which is really the motor circuit, which is what exemplified by the old definition of the disease. But there are also dorsolateral prefrontal and lateral orbital frontal cortex, and then the limbic and paralimbic areas. So we can define what happens in these areas when they become involved. So, for example, the orbital frontal cortex is associated with emotional and behavioral regulation. Dorsolateral and prefrontal areas are associated with executive impairment, and the anterior cingulate associated with motivation and initiation. And these parts of the brain, they're not uniformly invariably affected in ALS. We define ALS by the involvement of the motor system. So a diagnosis of ALS cannot be made without the motor system being involved, but a proportion of patients will have manifestations characteristic of these other circuits as well. And we know this because we've undertaken a series of population-based studies, and we know that this dichotomizes into two subgroups, those patients who on clinical presentation and with very detailed neuropsychological assessment have no evidence of cognitive change. The evidence is primarily of motor system degeneration, and the other 50% who have evidence of cognitive and behavioral change, and up to 1 in 10 of those patients, or 1 in 8 of those patients, will have frank frontotemporal dementia. They will present with the dementia in association with their ALS. A significant subpopulation also have language impairment. In terms of the behavioral changes, we see many different behavioral changes, and we can map those onto the different circuits that we know to be involved within the central nervous system. So this is a complicated condition. It's not a pure motor system, and we can attempt to cluster patients into different groups based on this simple presentation of the presence or absence of cognitive change and the age of onset, the nature of onset, whether it develops in the spine or whether it starts in the speech and swallowing muscles. So bulbar being speech and swallowing, and spinal onset being in the limbs. And we can separate patients into 3 clusters. An old paper that we published back in 2015, showing the different clinical trajectories of these different clusters. So it's a hierarchical cluster. You can also do latent clustering, which shows something similar, 4 groups actually, that can be further dissected out. But the presence or absence of cognitive change is a significant discriminator. So those with cognitive change, particularly executive dysfunction, have a much more rapid trajectory. So that's a different subcategory of the disease. We also know that if we look at the pathology of those with ALS, that the vast majority of patients have deposition of this protein TDP43 in the cytoplasm where it shouldn't be. It's a TDP43 mostly lives in the nucleus and it shuttles across, but in ALS it shuttles across and remains in the cytoplasm, becomes misfolded, and much of the pathology around ALS is thought to relate to this misfolded protein of TDP43. And ALS and FTD are linked. Frontotemporal dementia. So frontotemporal dementia is a condition characterized by degeneration of the frontal and the temporal circuits. And about 50% of those with frontotemporal dementia also have TDP43. And the pathology of those with ALS and FTD is that of TDP43 deposition. Smaller percentage of people have deposition of the protein FUS, and there are mutations associated with ALS that are in the FUS gene. So you can see there's a continuum from clinical point of view between those with ALS, pure ALS, and those with FTD. And the pathobiology is somewhat similar, but the clinical manifestations are different, and the reason why that is aren't very well understood. We know that ALS is a gene-environment interaction, and when we look at our population-based registers, which I'll speak about in a minute, we can divide the progression of ALS, or the development of ALS rather, into six steps. One of these steps is clearly the genetic load with which we are born, and we know from our population-based studies and also genomic studies that at population level, this accounts for about 50% of the risk, and the other remaining 50% is a gene-environment interaction. If an individual carries a single gene of major effect, the steps that you need to develop the disease go down to maybe 3 or 4. So SOD1, which is the first gene that was identified, there are 3 more steps that are required to get the disease, to develop the disease. C9orf72, there are 4 more steps. So even these single genes of major effect, there is a potential to carry this variant and not have the disease if the environmental factors that interact with that genomic signature are not conducive. So that's a very big complication or unknown aspect of this disease. We also know that there are more than one gene. There's more than one gene associated with ALS. I've mentioned 4 there. There are 4 major genes in Northern European populations and US populations. Those are SOD1, TDP43, C9orf72, and FUS. But there are many, actually, probably more than 50 genes now that have been identified and have been associated either as single genes of major effect within particular populations, or as genes that are at-risk genes, or that might modulate the clinical phenotype. For example, ataxin-2 modulates the clinical phenotype, And we know that ataxin 2 interacts with TDP43 deposition. And so ataxin 2, even though it's a modifier gene, is a target for gene therapies at the moment. Similarly with UNC13A, which is a modifier gene, but important in the pathophysiology of the disease. What do we know about the pathology of the disease? Well, the pathology of the disease, the pathobiology, has been worked out using an animal model. The animal model is that of SOD1, the SOD1 mouse, which was first developed in 1994. And much of the biology of what we understand of ALS has been established through extensive analysis of the SOD1 mouse. But there are many limitations of the mouse in terms of its translation to human disease. For one thing, SOD1 is not associated with TDP43 deposition, so the pathways are a little bit different. But from that, we can build a series of models that link to the various different genomic variants as well that might lead to potential targetable pathways for human disease. And the human disease, I've put a picture down on the bottom right of the what the human disease looks like with ubiquitination to inclusions containing TDP43 and selective degeneration of primarily the anterior horn cell, but involvement of other pathways as well. So we know that there are many different pathways based on different genomic variants that we can deduce from the function of the gene, and we know that there are many pathways that we have deduced also from the SOD1 mouse. But translating that into a new therapeutic modality has been challenging, and most of the drugs that have undergone trial, and there's over 90 now, have fallen into the valley of death. This is where the preclinical work and maybe early clinical work has been optimistic and suggestive of efficacy, and then the later clinical trials, which are very expensive, have failed. So moving from the mouse model, where most of the therapeutic initiatives up to quite recently have been in the mouse model. There are other models now, particularly high throughput screening using iPS cells. But there are arguments about doing that as well, in that much of what's happening in ALS is probably at network level as well, and it's very difficult to model networking within an IPS system. So we have a problem with the translation from what looks very therapeutically valid in the preclinical and even in the early clinical setting with small numbers, through to multiple failures in late clinical. So we have one drug that's licensed across the world, Riluzole, and we have 2 additional drugs that are licensed in the US, but not approved by the European Medicines Authority, the EMA, and are undergoing a phase 3 study at the moment to determine efficacy in a larger patient cohort with a much longer clinical trial duration. So those 2 drugs would be Relyvrio from Amylyx and Radicava from Mitsubishi. And it remains to be determined in larger phase 3 case control studies whether those drugs are indeed efficacious or not. So we have a rather poor track record in moving from preclinical to clinical. Many of the reasons, there are many reasons for that. Many of the reasons relate to poor trial design in animals, and also poor trial design in early human studies, and needing to go back and look at better trial designs and better outcome measures. And Ruben van Eyck probably is one of the world leaders in ALS in this regard now as a clinical trial statistician. But one of the things that we really have to understand is the heterogeneity of the disease. And there's no doubt but that ALS is a heterogeneous disease. And it's very unlikely that we'll have a drug that works on all forms of ALS. If we think about ALS as many factors that lead to the risk of developing disease, and then as the disease develops, may well be in the early stage we can modulate the clinical progression. But once the pathways are initiated of neurodegeneration, it becomes very difficult to stop this. So we need to identify the subgroups of patients at a very early stage where we perhaps can intervene with disease-modifying drugs that are targeted to the different pathogenic mechanisms that underlie these different clinical presentations, and identify this at a biological level. So ALS is a complex, heterogeneous human disease. It's a rare disease, but like all the rare diseases, it's probably a complement of a number of different pathogenic mechanisms that are recognizable, because that's what we've been taught to do by Charcot as a motor system degeneration. And modeling the disease in animals is probably not very effective, because we can model particular biological pathways in many different animal models, in the mouse, in the worm, in the fly. But this is a human disease, and there isn't any naturally occurring animal model of this disease in all of its iterations, because it's a complex disease of the complex anatomy and neurophysiology of the human brain, and that's one of the things that we need to understand. So moving from that, I'm a member of the TRICALS consortium. So TRICALS is the treatment initiative to cure ALS, and we're a group of centres around Europe that engage in clinical trials, but that are frustrated by the failure of clinical trials to date, and that are that have undertaken the commission, I suppose, to improve how we design clinical trials, but in doing so, to improve our ability to subphenotype the patients with ALS into those different subgroups that we might be able then to present for clinical trial of different compounds with different potential mechanisms of action. So a number of years ago, we wrote a paper called The Roadmap for New Drug Discovery, which had 5 lanes in it. And this was to develop a scale, to develop an N that's sufficiently large to enable us to study the populations at scale, and then to understand the different types of biological investigations that we can undertake that could be developed as biomarkers of the disease, so that we could develop new clinical trials for different patients, different subgroups, but also in doing so to develop this platform that we could then use to collect data and coordinate studies, and then improve the outcome measures that we use. So there are many different tasks here. And so this is really a precision medicine based approach. So this is to tailor the medical treatment to the individual characteristics of each patient. So to be able to classify people into subpopulations, different susceptibility, the biology and the progress of disease. And of course, this has been very successful in cancer, a combination of the ability to develop biomarkers of cancer, the ability to look at the cancer biology in real time through biopsy or through culture, and of course the understanding of the interplay between what's happening at a neoplastic cell division perspective and the interaction with the immune system. And much of the really enormous success within cancer, of course, has been in our understanding of the subtypes of cancer and their interplay with the innate immune system, and that's something that is beginning to evolve in the ALS field as well. So our precision ALS is to try and meet those objectives, is to try and develop a system by which we can undertake a precision medicine approach towards this disease, but this platform or this approach could be used for any rare disease. So this is transferable as a conceptual framework and as an infrastructure in the longer term. So I work in Dublin in Ireland, and I my academic affiliation is at Trinity College in Dublin, and the funding structure in Ireland is such that we have the Science Foundation Ireland, which is the main funding structure in the country for science, funds large centres. And I'm a member of 2 centres, one of which is a centre for rare neurological diseases called Future Neuro, which is based in a sister medical school called the Royal College of Surgeons, and a member of the ADAPT Centre, which is a large data science, computational science, artificial intelligence centre, so data scientists and computational scientists. So I'm a member of both groups, and my my thesis was that if we were to bring together clinician scientists like me, biologists, but mainly clinician scientists who see patients, and data scientists who would help us to construct a bespoke system of collecting, collating, and interrogating data from patients, we would be able to provide a novel mechanism by which we could really provide a precision medicine approach towards new treatment. So we're funded by Science Foundation Ireland to an academic industry collaboration where we have a number of industry partners, and the money that the industry partners provide in a co-created research set of projects is matched by Science Foundation Ireland. And my partners within the TriCals group are these sites here in Europe. And these sites were selected advisedly. So there are 42 sites from within the TriCals group. But the sites that are listed here are sites that have already developed the infrastructure to identify patients at population level. So we have population-based registers. So our register in Ireland has been running since 1994. And we have full surveillance of the disease and have done so since 1994. We capture everybody in Ireland who has ALS. And we've published many papers on this. And we've been able to show the information creep that's happening within ALS and our understanding of FTD, and the increasing incidence and prevalence partly related to the information creep, but partly relation to other aspects epidemiologically that are happening within the country around the risk of developing disease and the interaction between different cohorts and ages and the development of disease. So we're very skilled in this regard, as are our colleagues in the Netherlands and in Torino and in the UK. And there are clinic-based populations in Leuven, which effectively are almost population-based in France, in Tours, and in Barcelona, and a population-based register in Sweden and Scandinavia. So we've come together using the infrastructure that we have to identify patients. And our mission is to develop a system by which we can identify the right patient for the right drug for the right patient in the right dose at the right time. So the objectives of Precision ALS, as I said, transferable to any rare disease you might want to think of, is to enable the integration of multimodal, multi-sourced data from as many iterative processes as we can, not just clinical, the work I do, but imaging, and what we'll call wet biomarkers, genomics, and other new technologies relating to the brain, such as neuroelectric imaging or neurophysiological imaging using quantitative EEG and cortical muscular coherence and macro EMG. So looking at all of the different aspects of the structure and function of the brain in the context of this disease, and then collecting all of that, and then developing a new system of integrating all of these data on a platform at the next level up with machine learning and artificial intelligence. So that aspect of the platform is disease agnostic, really. It's using large data and looking at, in a latent manner, looking at the number of different types of clustering that we can undertake in these patients to identify common sets of attributes across these patients that might either signal a different clinical trajectory, a different pathobiological process, a different set of markers that we might be able to identify. So ultimately the aim is to generate new knowledge to derive translation and clinical research in neurodegeneration. So this is what the platform looks like. So there are a number of different work packages. Work package 1 here is the platform, and work package 7 is the machine learning and AI. And we've divided the other work packages into the types of data collection that we would undertake. So I'm going to show you a little bit of data to show you that even in rare diseases, it's possible to do this, and it's possible to do this at scale. So work package 1 is the development of the platform, handling this multi-source, multi-format, multimodal longitudinal data that's easily accessible, and then subject to appropriate data governance and control. And this is what it's going to look like. This is the data flow within the platform. These are the inputs. This is how the data is captured, the data are transformed, the data are analyzed, and the data are exported in either visualizations or in different types of export suitable for types of machine learning that we might want to undertake. So we've already developed a clinical tool to collect information on patients, a clinical tablet. so we can do large complex data capture in a clinical environment. We're also at this point now building the platform that will integrate data across the clinical domains, but also the other domains as well that I mentioned, and developing a system for retrieval and analysis, and then having an overarching data governance and data security mechanism, particularly in Europe. This is very important, obviously, from a GDPR perspective, but from good governance, it's important regardless of where we live. And so in the process, the project has been running for a year and a half. We have been collecting data across Europe as in consortia over the years, and the consortia are listed there on the slide. So we have large data sets that we've never really collated together before. We've been subject to the newer regulations on GDPR, so we've had to generate a whole new set of data sharing agreements in order to enable us to collate all of these data together. But we've been able to do this more recently, and we have a very large data set of data we've already collected to show proof of concept that this is possible even for a rare disease. So we've 21,800 patients on our extant data set. This is patients from our registers and from various different projects that we've done over the years, and quite a lot of granularity within this data set. And the types of analysis we've been able to do have been things like looking at the frequency of known genes. So these are rare. So, for example, SOD1 only accounts for a very small percentage of people with ALS, but we've been able to capture 400 or 500 patients with SOD1. We've been able to capture almost 1,000 patients carrying the CNNR72 repeat expansion. And we can start looking at the clinical characteristics of these patients. We can start looking at their clinical trajectories, the change in the clinical course of the disease, showing that, in fact, SOD1, which is considered a very rapidly progressive disease in the US, has quite a different clinical trajectory in Europe because the gene variants are different. Also, quite a bit of spread around the CNNR72, the gene with the least good outcome are CNNR72 and FUS, which are relatively rapidly progressive diseases. So this is a surprise, actually, because we would have always thought of SOD1 as a very rapidly progressive disease. We can also measure the rate of decline using one of the measurement scales that we have, and we can do this at scale as well, showing that the rate of decline changes as between year 1 and year 2. So in other words, the scale is not linear, and we can show the rate of decline based on the clinical trajectories of these patients with these different gene variants. So, for example, over 100 patients with SOD1 and 500 patients with C9orf, but actually we have a larger cohort of these patients that don't have the full data set. But even within this extant data set, we've been able to pull out really meaningful data for these rare diseases. And this is time to event for these various different genomic variants that we have, so we can do this type of analysis as well. So time from data from the time the patient is born to the date that they develop their disease, and this is quite a bit of variability, and then from the presentation to their disease trajectory. And we can also do things like looking at the healthcare ecosystem, looking at the cost of the disease, the burden for family members, and out of that, building a complete cost of disease. So even with the extant data, using these population-based data sets, plus some of the data we've already collected, we can undertake really meaningful analyses. And this is the first stage, and we can start undertaking the machine learning even with these relatively incomplete data sets. But we're going to have a much more complete data set going forward. We're aiming to collect more than 6,000 cases in the first 3 years, because we already have other sites who wish to join the 9 sites that we have. So we have our consortium agreement just about signed, and we will be inviting other sites to join now in the next month or so. So our prospective data collection is going to be collected in real time, close to clinic visits. We'll be incorporating service utilization data, and it will be population based. And this is an example of the data collection tool that we have. You can see that we're collecting demographics, we're collecting clinical data, we're collecting progression data, we're collecting socioeconomic data and service utilization data. So these are going to be very, very rich data sets. And we also are building a new program where we're collecting these different types of biomarkers, which will be incorporated into the patient data platform as well. And this is types of examples of biomarkers that we will be looking at We already have a program running with proteomics and with neurofilaments as well, and we'll be developing. We have a program running with imaging and with neurophysiology as well, and we'll be collating all of those data, so that we can really incorporate these into the clinical applications of biomarkers that will be bespoke, but possibly for an individual subtype of disease, or that could be a biomarker of disease progression. There are many different types of biomarkers that we could use here. We're also we've got a program of digital outcome measures. So the objectives here to design and test new measures to incorporate these into harmonized footwear collecting, to develop APIs for selected apps, and then to identify the barriers to using digital collections. And so we're in ALS. These are the types of constructs that we're looking at, but this could be in other rare diseases. You would possibly have other constructs as well. There are matching apps to each of these constructs as well. From a patient journey point of view, this is what we're looking at. We can identify patients in our population data sets from the day they're diagnosed, and then we can go back to a pre-diagnostic journey. And we've done this in 500 patients already, showing that you can track back to the first symptom, the pre-diagnostic journey, and map that out and cost it as well. So we're already able to do that. Going forward, we then collect the entire patient journey. And that's, of course, informed then by these touch points and more detailed data. We can do a lot of data mining, and then we can start modeling the from a health economic point of view. And this is some of the modeling work that we've already done here. Again, this could be used in any rare disease at scale. The problem is it hasn't been possible to scale, and that's what this machine is going to be able to do. And then finally, the machine learning part.
Work package setup is really
about. Oh, sorry, don't mind that. Sorry, there shouldn't be a speaker there. Don't mind that. So we're looking at cohortization and insight generation, and moving from current situation to future. There's a speaker there that shouldn't be there. And then so ultimately AI machine learning will be cohortization, disease progression modeling, insight generation, and then the potential for developing the types of placebo groups that Ruben will talk about, developing digital twins, developing avatars, being able to identify patients within the real live machine that we could then use as a control cohort for patients. So obviating the need for placebo groups in trials as the model moves forward. So that's really what I wanted to talk about. I'm happy to take questions. That's a website for what we're doing. But I think this is a machine that we would be suitable for any rare disease. And I think it is possible to collect information on rare diseases at scale with the right infrastructure. Precision ALS, the plan in the longer term, is that the shoe leather that we spend on collecting the types of clinical information that requires research nurses and research assistants, we will have all of this automated by the end of this program. So the digital tool that we have will ultimately plug into the EPRs within the various sites. There'll be a lot of capability to upload data from existing data sets, so that the need to fund people to go around collecting data, if there's an infrastructure within a particular center, we should be able to upload those data using the existing infrastructure that's there. So I think this is the way forward for rare diseases, and I think Precision ALS is an exemplar of what we can do with rare diseases moving forward. So I'd be happy to take any questions. Thank you very much for listening to me.